Hi everyone, and welcome to the second part of this presentation. My name is Michael, and I want to start off by saying thanks for having me. I'm very excited and very honored to be part of this very first Asia-Pacific working group. I'm going to just jump straight in and carry on from where Peter left off and talk more about how genetic tools can be used to answer important questions in sea turtle biology and conservation. Now, I've spent a great deal of my career thinking about this one, which nesting stocks use which feeding grounds. So that'll be the main focus of what I talk about today. So we've been trying to understand connectivity since the first tagging studies more than, I think, half a century ago, if not more. Studies have used flavor tagging or pit tagging and satellite telemetry and stable isotopes to look at connectivity. But genetics, I think in particular, has been one of our best tools for quantifying connectivity between rookeries and foraging grounds for sea turtles. So how do we characterize connectivity using genetics? Well, in the first part of Peter's talk, Peter's presentation, we talked about the importance of identifying stock structure in order to better manage and protect rookeries that are genetically distinct. And the other reason why that's important is because by characterizing these management units, we create a reference of rookery baseline data that we can use to look at connectivity between rookeries and foraging grounds and be able to define the geographical boundaries of each stock. So there are essentially two approaches that are used to assess connectivity using genetics. There's mixed stock analysis, MSA, also sometimes called genetic stock identification, and there's assignment tests. Both are based on the principle of comparing the genetics of samples to a reference baseline so that we can assign population origin. But the two approaches differ in a few significant ways. I think most importantly, mixed stock analysis can be used with both mitochondrial DNA and nuclear DNA. And because mitochondrial DNA can be compared across studies, we have a good baseline data available for most regions and species. Assignment testing, on the other hand, uses nuclear DNA, with microsatellites being the marker of choice up until recently anyways. And because microsats are difficult to compare across studies, we have limited baseline data available for assignment tests. So because mitochondrial DNA has been the preferred tool for defining rookery stock structure, it is a very useful tool for tracking the natal origin of sea turtles. And so that means that almost all the genetic information we have that's used for mixed stock analysis in turtles is based on mitochondrial DNA haplotype data. So this is essentially how it works. The simplest situation that we can think of is a mixture involving only a few stocks that have strong genetic structure with no shared alleles or haplotypes. So in this overly simplified example, we have three baseline stocks with fixed haplotype differences represented by the three colors and a shared feeding ground. Now in this example, it becomes pretty straightforward to trace back the stock origin of turtles in the mixture. We can assign turtles individually, or we can do a proportional estimate to say 80% of the turtles in the mixture originate from stock A, 50%, 15% from stock B, and 5% from stock C. Easy peasy, right? But I hate to say it, but fixed differences in haplotype frequencies like these ones are rarely, if ever, seen in marine turtles. So most often the baseline rookeries are distinguished by different frequencies of alleles or haplotypes. So this figure here shows the basic principles of a mixed stock analysis. And this is much more the case that we see in the real world. Right? So again, we have an example of three stocks with the pie charts now representing the haplotype frequencies in each stock and in the mixture. And as you can see, they are now differentiated based on significant haplotype frequency shifts. And they share haplotypes, so there's less genetic structure. Now that means that we can no longer directly link the origin of turtles in the mixture to a nesting origin based on individual haplotypes. Right? So in these situations, the best thing that we can do is to estimate the relative contribution of the baseline stocks to the mixed sample. And here a mixed stock analysis gives us a really powerful way to analyze our mixture data. And so what we end up with are results that show the mean relative contribution of each baseline stock surrounded by some level of uncertainty. I just want to quickly mention also that there are three criteria that are super important for an accurate mixed stock analysis. The first one is to have a comprehensive genetic baseline of the stocks, so the nesting part, that you think might be contributing to the feeding ground. The second one is to have strong genetic structure among the source populations. And the last one is to have a good sample size of both the baseline and the mixed populations. So it's very important to understand that the power of this approach really depends on how well these three criteria are fulfilled. And when they are, we get vitally important information about connectivity. But when they're not, it can really lead us down a path of incorrect conclusions. So you've got to be careful. Now, we won't go into any details of the mixed stock analysis in this talk because we're going to revisit this topic in much more detail in workshop 4, where we go into details about stock structure analysis and mixed stock analysis. So if you're interested in learning more about these topics, please stay tuned for workshop 4 at the end of April. I think for now, I'm just going to go through a few examples that use mixed stock analysis and really highlight some of the types of questions that it can help us answer. Okay, so my first example here is, as far as I know, the first study using mixed stock analysis in the Pacific Ocean and looking at where green turtles foraging in Hawaii might be coming from. So in Hawaii, green turtles forage primarily around the Hawaiian Islands, but the main nesting beaches are found at the French Frigate Shoals about 1,000 kilometers to the northwest. Right? So here, Dutton and colleagues used a baseline of stocks from across the Pacific and the mixed stock analysis to show that the turtles foraging in Hawaii all originated from the nesting stock at the French Frigate Shoals. And in fact, of the almost 700 samples that they looked at, they only found 2 turtles with haplotypes not found at the French Frigate Shoals. So that means that all the Hawaiian feeding grounds are completely isolated from rookeries outside of the Hawaiian archipelago. And it means that the Hawaiian green turtles can be assessed separately from other Pacific stocks with respect to risk and how this population is managed. Now, since then, that first study, now more than 30 foraging grounds and many, many more rookeries have been published using mixed stock analysis across Asia-Pacific, as you can see from this map here. So what we're learning from these studies is that there is a lot of variation in the results. Some foraging aggregations are composed mostly of turtles from the nearest stock, such as Hawaii, but it is much more common to see feeding grounds that include turtles from multiple regional rookeries, such as these ones from Malaysia and Fiji. So mixed stock analysis provide us with the knowledge about where turtles are coming from, which means that we can now include nesting beaches and foraging ground into things like risk assessments. So, as conservationists and managers, as I'm sure many of you out there are, we're often interested in knowing which populations are impacted by threats away from the nesting grounds. So this could be threats occurring in foraging grounds or during migrations from either fisheries bycatch or harvest or poaching. So again, using genetics allows us to identify the natal origin of turtles, which is crucial if we want to assess and compare impacts on regional rookeries. So a relatively straightforward example of fisheries bycatch are Pacific loggerheads that Peter showed earlier. Now, as you remember, the only two main nesting regions in the Pacific are found in Japan and the ones in Australia and New Caledonia. But we also know that loggerhead turtles undertake these amazing long-distance migrations to the Pacific, across the Pacific to rich foraging areas off the coast of Central and South America, where some of them get caught in coastal and oceanic fisheries. So here again, mitochondrial DNA from turtles collected from fisheries in the North Pacific, as well as those immature turtles that make it all the way to the feeding grounds in Mexico, show a clear link with the Japanese genetic stock. And the turtles that are caught in fisheries off Chile and Peru show a clear and exclusive link to the Southwest Pacific nesting stock. So we have two discrete units with no overlapping boundaries and a very clear result that we can use to plan appropriate conservation actions. Another great example is how we can use these methods to tackle the illegal trade in turtle products. Hawksbill turtles in particular have been and still are heavily exploited for their shell to make tortoiseshell products like these ones. So this paper is hot off the press, and the aim was to extract DNA, or see if we could extract DNA from tortoiseshell products sold at markets in Papua New Guinea and the Solomon Islands, in order to find out which nesting stocks the turtles came from. And so through magical work of Aaron Lacasella and Noah, we were able to extract and sequence 800 base pairs of mitochondrial DNA from products like bracelets and earrings. And so the results that we got indicated that about 70% of the products from these markets had haplotypes found primarily at the Solomon Islands nesting stock. And I think equally important, did not match haplotypes from nesting populations in Australia or Southeast Asia, which could then be excluded. But as you can see from the map, which shows the haplotype frequencies of the handful of rookeries that have been published in the region, we have an incomplete baseline for hawksbills, which means that we can't do a full analysis at this point. We also found that almost a quarter of the market samples had what we call orphan haplotypes. So these are haplotypes not found at any rookery to date. And that really highlights, I think, an important point when doing mixed stock analysis. Because these orphan haplotypes indicate that we either don't have an adequate sample size from rookeries or that they're unsampled rookeries. Orphan haplotypes provide a really important reminder that the baseline is incomplete and that we need more sampling. Still, I think with this study, we've really demonstrated an effective method for getting DNA from hawksbill products, and it shows great promise as a tool to combat the illegal trade in hawksbill products. Now, because this is such an important topic and one that has gathered a lot of interest in the region, we have a great session focusing specifically on illegal trade and forensics, and an exciting new project called Shell Bank in workshop number 6 in June. So again, stay tuned for more information on that workshop. Now here's another hot topic for marine turtle conservation, climate change. We all know that in sea turtles, the proportion of female hatchlings increase with the incubation temperatures. So with increasing global temperatures, many sea turtle populations are now in danger of becoming feminized. So I wanted to take the opportunity to show how working in foraging areas gives us an opportunity to look at impacts of climate change. So when we work in feeding grounds, we often find a mix of turtles of different life stages. So these turtles represent different cohorts or different generations of turtles. So the secondary sex ratios that we see at foraging grounds can give us information on the mix of immature and adult turtles that have been hatched from different nesting beaches over many years. And a few years ago, I was fortunate enough to be involved in a study on the Great Barrier Reef, whereby combining information on the stock origin using genetics and determining the sex of turtles from different size classes, we were able to calculate stock-specific sex ratios for each size class. And that really gave us a very unique window into sex ratios being produced from regional rookeries over the past many decades. And so from that study, we got some really interesting results that showed a moderate female bias, about 70% females, in turtles that were born on the cooler nesting beaches in the southern part of the Great Barrier Reef. But the turtles that have been born on the warmer nesting beaches in the northern part of the Great Barrier Reef were extremely female biased, with almost 100% of the juveniles and the subadults being female. So these results really sounded the alarm bells because they suggest that the northern Great Barrier Reef green turtle rookeries, which are some of the largest in the world, have been producing only females for more than 2 decades probably. So combining genetics and secondary sex ratios can provide a better understanding or new understanding of what these populations are dealing with. And I think importantly, it can show us what the breeding sex ratios are today and what they might look like 10 or 20 or 30 years from now when these young turtles grow up to become adults. And so to me, these are questions that we need to discuss. And I think in order to fully understand the impacts of extreme female biases, we need to learn a lot more about the male components of sea turtle populations moving forward. So with that, I want to say thanks for listening, and I'll pass the baton off to Peter to tell us his thoughts on male sea turtles, and to round off our presentation with some words of wisdom. So over to you, Peter.
Thanks, Michael. Well, I don't know about words of wisdom, but I did want to show how we've been using nuclear DNA to peek into the sex life of sea turtles. Earlier, we talked about how we can use genetic fingerprinting with nuclear DNA to identify individuals. For example, there's 3 questions we want to know about that involve genetic fingerprinting of males. One's the sex ratios of breeding adults, another's mating patterns, such as levels of multiple paternity, and by tracking males, we can determine their male reproductive site fidelity. But how can we assess the male component of the population without being able to actually tag or sample the males? Well, the answer is you can do this on nesting beaches by sampling all the females and their offspring. By comparing the mother's genetic profiles with her hatchlings, we're able to reconstruct the father's genetic identities. And this allows us to identify and count the number of males breeding in any given year. We've been able to do this with work that Kelly Stewart has been leading in St. Croix for a number of years, using nuclear microsatellites. In future workshops, we'll talk about how we're expanding this work to using SNPs and genomic techniques. But for now, I wanted to just highlight one of the surprising findings for this studies. We've found that sex ratios are pretty consistently 50-50 male to female. And this is surprising given that what Michael talked about, and given that the hatchling sex ratios are extremely female biased. So to Michael's point earlier, it's important to assess the sex ratios at all the life stages to be able to track impacts of climate change. This example shows how we can do that for the breeding populations. Before we wrap up, I do want to mention that there's a lot we haven't talked about, particularly the emerging technologies and applications. Many of you may be involved in some of these, for instance, environmental DNA, gene expression, and a whole suite of genomics application that are expanding our capacity to really get into many of the questions that we went over today. One highlight is this year we published a fully annotated high-quality sequence that's available for leatherbacks and green turtles. We'll explore these in all these topics in more depth in Workshop 5 at the end of May, and we'll talk about novel and alternative genomic tools. Finally, I wanted to say again how excited I am to be part of this network of sea turtle folks in Asia Pacific. I'm glad the turtles have brought us together. Many of the examples that we've spoken about today demonstrate the importance of working together across international boundaries to expand those genetic baselines for all of the species. In the coming workshops, we'll be learning much more about the genetics work being done in the region by all of you, and I really look forward to that. It's my sincere hope and belief that this working group will help establish or expand collaborations, train students, and build regional capacity and a network of field researchers and in-country laboratories to share ideas, samples, and data. On behalf of Michael and I want to thank you all for listening, and I look forward to discussing these ideas with you in the coming weeks, months, and even years.